A very good morning to everyone attending this webinar from Ancrum. This webinar will start exactly at 11:30. Till then, we will wait for other attendees to join in. So participants are joining in. I can see total 35 scientists have joined the session. So thank you for this very good response for this webinar. We will start this webinar by 11:30. We will wait till then. We will allow other attendees to join in. Then we will start the session. We will wait for 5 more minutes. Hello, good morning all. Hello, Ancrum team. I hope I am clearly audible and my screen is visible.
Yes, Vishwaji.
Okay, thank you. First of all, very good morning to all attendees. Welcome to the HPTLC webinar being hosted by Ancrom Analytical Specialist. In this webinar, we will cover different aspects of HPTLC analysis for phospholipids, and my Ancrom lab colleagues are also present in this webinar to answer your questions. So I request you to ask your questions in chat box or in Q&A, or you can raise your hand, or at the end of this session we are taking question and answers. You can ask at the end. So at the end we will unmute you. You can ask your questions live, and this session will be recorded for our quality monitoring purposes. And with this, I request you all to please feel free to give the opinion in poll. So we will start our polls right now, and I request you to please question the poll, and then we will start the session. Whenever you are filling the polls, meanwhile other attendees will join in, and we will still wait for 3-4 more minutes to join all attendees, those who have registered in this webinar. So I request you all, please fill the polls. If all attendees are joining in the session, meanwhile, I request all those who have joined, please answer the poll questions. Okay, so as I can see, yes, you are currently analyzing phospholipid excipients. So the attendees who are attending this session, out of that, routinely analyzing are nearly 22%, and planning to start using phospholipids analysis nearly 44%, And no, most of the participants, like 27% is no. Occasionally, some participants are doing analysis of phospholipids, nearly 9%. So 9 plus 19, that is huge number. Nearly more than 50% attendees are doing phospholipid analysis or planning to start with the analysis of phospholipids. That is huge number. So definitely this session will be very useful and fruitful for you. And in next 1 hour, you will see the HPTLC benefits for the phospholipids analysis. So thank you for attending. Thank you for joining and sparing your time. Now answer other questions of the poll. One more question in the poll. That is huge number. I have seen that more than 50% of the participants are already doing phospholipids analysis, or they are now planning to do that. Now, the second question, which technique does your lab primarily use? In that HPTLC, most of the participants, that is huge, means 60% nearly, and that polls are rolling. Mostly HPTLC is highest number, like 65%. Then LCMS nearly 11%, HPLC 22%. That are all huge numbers. But HPTLC is first choice, I think, as per this poll, and these numbers are changing now. But HPTLC wins here. Most of the participants are using HPTLC methods for the analysis of phospholipids as a primary method. Thank you for responding to our polls again. Okay. Thank you for attending the polls. From these polls, I got the idea exactly which kind of attendees have joined, and I'll try to focus more and more as per your requirements. Therefore, we have taken this poll survey. Again, starting with the session today, now we will start the session. Because now 11:38 in my clock. So in between, other participants will join in. Many participants have registered across India. So starting with the introduction of Ancrom, we are analytical specialists since 1978, and we have well-equipped lab at Mumbai. And in Mumbai, we are doing routine analysis on CAMAG HPTLC systems for different application fields. Out of that, phospholipid is our primary focus for our pharmaceutical customers as well as food cosmetics customers. Now, starting with topic of interest today, what are phospholipids and what is the requirement of its analysis? How we can achieve that analysis very in faster way, in cost-effective way, and simultaneously for high sample throughputs. So, what are phospholipids? So, phospholipids are a class of lipids that are crucial components of all cell membranes. They have a unique split personality structure that makes them incredibly useful in both biology and pharmacology. So one end of this phospholipid is polar in nature, that is hydrophilic, and one end is non-polar in nature, that is hydrophobic. So it is very useful to form lipid bilayer. So formation of lipid bilayer is very useful in pharmacology as well as in biology. So basically, one end is polar and one end is non-polar, makes phospholipids amphiphilic. So this unique nature, amphiphilic nature of phospholipid molecules plays crucial role and increase its use. in different application fields, mostly in pharmaceutical, and it is very useful for drug absorption. Now, why phospholipids and their impurities analysis is very important? Because if any impurity generates in the phospholipid, that directly impacts the stability of your product, efficacy of your product, as well as it can fail regulatory compliance of your product. So during product manufacturing, mostly as an excipient, you are using phospholipids, and if any impurity detected in that excipient, it is directly hampering the product quality, its stability, efficacy, and ultimately it results in regulatory compliance failure. So different strategies we are using to analyze impurities of phospholipids. Now the question arises, how impurities are coming in the picture, how impurities are forming along with major main API substances of phospholipids. So this is the process of manufacturing of natural phospholipids. Natural phospholipids are mainly manufactured from soya beans, sunflower seeds, or egg yolk. And then crude oil extraction. After crude oil extraction, water degumming process carried out, and in after that, acetone fractionation is the main step. And during acetone fractionation, de-oiled lecithins, that are phospholipids, are separated from oils, and oil dissolved in acetone. So during this step is very important. During this acetone fractionation, water de-oiled phospholipids are separated, and we can say it as natural phospholipids. Means phospholipids manufactured through natural sources like soya bean, sunflower seeds, or egg yolk. And further, its purification carried out by using different column chromatographies, long columns filled with aluminum oxide, etc. And after purification, we are getting completely pure uh, natural phospholipids. And in that, this PC, phosphatidylcholine, phosphatidylethanolamine, etc., are different types of phospholipids. As you can see on your screen, phospholipids are somewhat waxy and solid in state, nearly white to off-white color. And this is the second type of phospholipid and manufacturing process for synthetic and semi-synthetic phospholipids. In natural phospholipids, there are some limitations. They don't have a particular carbon chain and selective carbon chain structure. So a synthetic, semi-synthetic phospholipids gives us to embark the exact composition of carbohydrates, that is polar and non-polar chains of carbohydrates into the molecule. So during the manufacturing of semi-synthetic and synthetic phospholipids, again some impurities, we can say it as process impurities, or after generation minor products during the organic synthesis, all these impurities may be hazardous and decreases the stability of your final product. And as an excipient, if we are using phospholipids, if any impurity coming along with the main phospholipid, it may hamper the final product, it may damage the final product. So definitely there is a need to check the impurities present in phospholipids, either from natural source manufactured or from synthesis manufactured phospholipids. Both are very valuable in industries. Naturally obtained phospholipids are slightly cheaper in prices, but uh, possibility of impurity generation is very high. But synthetic and semi-synthetic are very accurately made and synthesized in industries. But again, the possibility is very less. But if any small amount, any small fraction of any impurity enters into the final product, definitely it will decrease the quality of phospholipid. So these are 10 major impurities detected in natural phospholipids and synthetic phospholipids. In natural phospholipids like lysophosphatidylcholine, that is common in synthetic phospholipids also, lysophosphatidylcholine. So there are many methods available right now. Out of that, we have selected three methods. And why to separate and quantify phospholipids and their impurities? Because phospholipids are critical building blocks for advanced drug delivery, like liposomes, micelles, and emulsions. And accurate separation and its quantification plays very vital role during encapsulation of formulation. It plays vital role to keep control on its quality. And in the industry of food and nutrition and health, the food and nutraceutical manufacturers precisely quantifying main substance, main phospholipid, along with their impurities, because legally it is very important. Legally it is required to verify and declare the detected impurities in the phospholipid used for the product manufacturing. Currently, these three important methods are available for the analysis of phospholipids and its impurities. First method, as Paul said uh, during starting of this webinar, we have taken a poll. In that, we understood that many participants are using HPTLC method for the analysis of phospholipid. So HPTLC is the first choice. Why? Because highly matrix robust, disposable stationary phases, and high sample throughput up to 15 to 20 samples we can analyze simultaneously. Reproducible and sensitive results we can achieve, and very cost effective, 50 to 60 rupees per sample analysis, and fully compliant with regulatory, current and latest regulatory. Then HPLC or UPLC. But the problem with this technique is its column clogging issues due to polar and non-polar nature of different phospholipids, and its high cost. It cost you nearly 200 to 300 rupees per sample, but it is fully compliant, and further sensitivity related things are better in UPLC and HPLC. And last technique, third technique, is very commonly used for phospholipid screening, large sample pool screening, is TLC, thin layer chromatography. So it is based on only visual observations and good for fast preliminary screening, and it is coming under non-compliance. So this table shows you that HPTLC is the first choice for the phospholipids analysis, and this is the complete system, CAMAG HPTLC system for phospholipids analysis. This provides you the complete step-by-step instrumentation in HPTLC workflow with fully compliant environment, 21 CFR Rule 11 compliant environment in CGLP, CGMP laboratories. We can use this CAMAG HPTLC instruments and carried out routine phospholipids analysis. So analysis of phospholipids starts with stationary phase selection. So there are different kinds of stationary phases available in the market. Based on the need of the method, we have to select a suitable stationary phase. Mostly we are using polar stationary phases with silica coated on aluminum backing or glass backing material. These plates are always new, so 100% new stationary phase we are using for each new analysis, so there is no possibility of cross contamination. Then after selection of suitable stationary phase, we have to apply samples and standards of phospholipids on HPTLC or TLC plates. For that, CAMAG HPTLC applicators we are using, automatic TLC sampler for high sample throughput sample application on plate, and Linomat 5 semi-automatic sample applicators. The Linomat 5 or ATS4 uses a spray-on technique to apply samples as narrow bands. So generally in manual type of TLC, we are applying samples and standards in the form of spots. But by using HPTLC applicators, we can apply exact volume of sample and standard in the form of bands. So band-wise application with spray-on technique plays a crucial role for increasing the quantification accuracy in phospholipid analysis. Then in the HPTLC workflow, next step is chromatographic development. For chromatographic development, we are using CAMAG twin trap chambers or humidity control chambers like ADC3, automatic developing chamber with humidity control. It consumes only 30 to 40 milliliters of mobile phase to develop 20 by 20 or 20 by 10 centimeter plates and to analyze 12 to 15 samples simultaneously on a single plate. With a rapid plate development time just up to 10 to 15 minutes, this method drastically minimizes solvent consumption. So it is 5 times lesser solvent consumption compared to other conventional techniques like HPLC or other techniques. So it reduces 80% use of solvents. So generally HPLC uses 250 to 300 milliliters for 12 to 15 samples, but it uses 30 to 40 milliliters only. Now, in the phospholipid analysis, important step is derivatization because phospholipids typically lack strong chromophores. So without derivatization, phospholipids are not visible with UV visible detector or fluorescent detector. So making derivative of phospholipid is very important and we need to carry out that on plate. So there are two important methods for derivatization. Dipping the plate into derivatizing reagent, that is copper sulfate reagent, most of the time we are using for selective derivatization of different phospholipids and its impurities. And after derivatization of phospholipids, easy detection is possible because of homogeneous and user independent derivatization by using CAMAG immersion device for dipping the plate into reagent and CAMAG derivatizer for spraying the derivatizing reagent on the plate. After that, we need to heat the plate at 170 degrees Celsius for 10 minutes on CAMAG plate heater. CAMAG plate heater provides a uniform and homogeneous heat from the bottom of the plate and accurately forming derivative on plate in situ. So it increases 100% reproducibility during derivatization, plate to plate, lab to lab, analyst to analyst, and it is 5x faster, 5 times faster than other conventional derivatization technique like manual spraying, manual dipping. And unlike TLC, it is not depend upon user. It is user independent derivatization. So up till now, we have seen different steps like sample application, chromatographic development, then derivatization. And after derivatization, we can scan the plate under scanning densitometer. And scanning densitometer provides you the spectral range of 190 to 900 nanometers for exact and selective quantification of phospholipids. Generation of 510 nanometers, 510 nanometer wavelength with tungsten lamp is very important, so that we can do with scanning densitometer and generate chromatograms. Here I have given you the example of separation of different polar phospholipids in the form of chromatogram on your screen. You can see uh, at the bottom. So CAMAX scanner scans the plate and generate chromatograms 5 times faster than other column chromatographic techniques, and it is faster than other detectors. And after complete analysis and after photo documentation, the next step is automatic calculations. So CAMAG HPTLC system along with its 21 CFR rule 11 compliant software provides you the automatic calculations and report generation steps. So calibration and data processing is very easy. Generally, I have seen in phospholipid analysis, Michaelis Menten one regression mode is recommended. So we can utilize that mode also. There are 5 regression modes to check the linearity of the standards phospholipids or their impurities. Linear 1 response, linear 2 regression mode, polynomial, Michaelis Menten 1 and Michaelis Menten 2. In all these regression modes, if you can see on your screen at left hand side, the response of different phospholipid and their linearity reported in linear 2 regression mode. It is giving very good R value and CV value. And at right hand side, you can see regression selected Michaelis Menten 1 for the same linearity analysis. Here also we got a good CV and R value. So software will automatically plot the linearity curve and do quantification of different impurities from phospholipids, and software will automatically tell you the percentage of particular impurity in your main phospholipid. Now, starting with some examples of phospholipid analysis done by CAMAG. This is one method I am explaining for analysis of mostly polar phospholipids. Here you can see lysophosphatidyl-choline, sphingomyelin, and phosphatidylcholine, phosphatidylserine, phosphatidyl-inositol, and phosphatidic acid, phosphatidylglycerol, and phosphatidylethanolamine simultaneously separated on a single plate with a single run in isocritic mode in mobile phase. So this is very fast, rapid method developed by CAMAC. And in this method parameters, CAMAG has used HPTLC silica gel plates and developing solvent as a cyclohexane, propanol, and water. These are green solvents, and derivatization again with copper sulfate reagent. Now, why to choose this CAMAG HPTLC method for phospholipid analysis? Because it separates 9 lipids simultaneously on a single plate, and it is multi-industry versatility tailored to support formulation and development like liposomes, micelles, emulsions, different formulations in the pharmaceutical and cosmetic sectors. It is having high sample throughput, so simultaneously 9 different lipids that are impurities of phospholipids we can detect on a single plate for many samples together on single plate, and eliminate sequential injection bottlenecks, so reducing the time of analysis, elimination of hazardous solvents like chloroform, etc., and replacing that hazardous solvents with greener solvents like cyclohexane. Rising market demand, as I seen in the poll also, and during our application support we are visiting pharmaceutical customers. There is a demand from the pharmaceutical industry, mostly cosmetic and food industry, for phospholipids. And to to give something for this demand, a rapid solution, cost effective and fast solution, HPTLC plays vital role. So this is another method, second method for the analysis of non-polar phospholipids. In this method, as you can see at left hand side in the plate image, there are 9 tracks in front of you, and in track number 1, 2, 3, all the polar phospholipids are remaining at the bottom at lower 0 RF value, that are N-acyl phosphatidylethanolamine, phosphatidylethanolamine, and lysophosphatidylcholine. These are 3 polar impurities, phospholipid impurities remaining at the bottom, not rising up with this mobile phase, because this mobile phase is non-polar in nature, and developing only alpha tocopherol, stearic acid, and tripamidine. That are 3 non-polar impurities in the main substance. So main substance of phospholipid is in track number 9, remaining at application position, and there are no non-polar impurities into that ninth number as such sample track. So no impurity like alpha tocopherol, stearic acid, or tripamidine detected in HSPC at such sample as per this image shows. So these kind of visual observations are very easy and faster in case of HPTLC. HPTLC offers this benefit of photo documentation and to observe the actual separated phospholipids and its impurities on plate, whether it is having polar in nature and non-polar in nature. uh, the mobile phase we can develop will be versatile to separate all. So HPTLC method for the analysis of phospholipids, as I show you earlier in this plate image, for development of this plate, we have used this mobile phase, and stationary phase is again common, that is HPTLC silica gel. So as you can see, every time we are using similar specifications of stationary phase, but we are changing only the composition of mobile phase depend on the polarity. Now, why to choose the Enchrome HPTLC method for phospholipid analysis? Because it is useful for comprehensive phospholipids, successfully screens and quantify. It is not only for screening, for visual observations, further quantifies all major lipid classes, including tripalmitin, stearic acid, etc., non-polar lipids also. High sample throughput, parallel processing, 25 to 30 samples we can carry out in 2 hours simultaneously. So only 4 to 5 minutes are required for every single sample analysis. And these all methods are available in our lab. I have shown you two important methods prominently used in the industry, which are fully validated as per ICH-Q2 R2 guidelines, and uh, tailor ready made for installing in your lab and to carry out your work under 21 CFR Partillion compliant environment. This is again one photo documented plate image where we have analyzed 10 samples of phospholipid HSPC, hydrogenated soybean phosphatidylcholine sample, 10 samples together on a single plate simultaneously for the detection of lysophosphatidyl phosphatidyl ethanolamine, N-acyl phosphatidyl ethanolamine, and stearic acid here. So all four impurities of this main HSPC phospholipid are separated at different RF value, as you can see on your screen. And out of that four impurities, only one impurity, that is stearic acid impurity, detected at the same RF value of the standard in all of the 10 samples, and other impurities are not detected. Now, if we use scanning densitometer further after photo documentation, we can easily quantify exact percentage of the stearic acid impurity detected in HSPC sample. This is another plate for actual sample analysis done at Enchrome laboratory by using CAMAG HPTLC system. Here we have applied three main HSPC samples, hydrogenated soybean phosphatidylcholine samples. This is one kind of phospholipid useful in the formulation industry for tablet coating, and in that we have not detected here phosphatidyl ethanolamine and N-acyl phosphatidyl ethanolamine. We have applied together along with the samples, but samples does not contain any of this impurity, but we got very good linearity for impurity detected. So we can do, if any impurity detected in the sample, we can do its sensitive quantification. Now, coming back to our topic of discussion today, how HPTLC offers you faster solutions, 5 times faster solution. Because if you see this comparison for per sample analysis time, it is very faster, 5 times faster than traditional HPLC or TLC. Then streamline method development and validation for R&D. So method development validation is again very fast. Generally for HPLC, it will take several months to develop a method and validate. But in HPTLC, we can develop a method and do validation as per ICH-Q2 or to latest guideline in 3 to 5 days. So it is 5 times faster. Cost per sample, it reduces the cost if you use HPTLC 50 to 60 per sample only. So it is 5 times lesser cost than traditional HPLC. Now, these HPTLC methods and these analysis methods, as I discussed during my session, are very useful to pharmaceutical products manufacturers, food and cosmetic products manufacturers, regulatory control laboratories or testing laboratories, government as well as private sectors. Then herbal and nutraceutical product manufacturers, those you are using phospholipids for formation of bilayers or, or an or an excipient in their final products. So with this, you are gaining 5 times faster efficiency, 80% solvent reduction. So we can say HPTLC is very green technique for the analysis of phospholipid, very rapid technique, and 100% compliance ready. As our software, VisionCat software and complete system is 21 CFR Part 11 compliant. So we can generate analysis reports in very faster way. Then why Enchrome? We are technologists, not traders. Since 1978, we are in this industry. Now, with this all slides and presentation, I wanted to show you some live examples as we did in our laboratory, and I will show you some VisionCat's analysis files where we did actual analysis of phospholipids. I'll open the CAMAG VisionCat software, that is HPTLC system control software, and in that I'll show you the analysis for impurity detection done in our laboratory and report generation, and it's 21 CFR Part 11 compliance. Hello, Ancrom team. I hope my screen is visible clearly.
Yes, Vishwajit.
Okay, thank you. So this is the CAMAG VisionCat's software for HPTLC system control, and in this software we can save all the data files in folders uh, in SQL database. After entering your login details, username and password, this first window will open up, and on your screen, as you can see, I have made a separate folder for the data of phospholipids analysis. I'll double click on this data folder, and in that I have selected 3 examples analysis files. Out of that first example analysis file, I will open up. Here we have checked the linearity of N-acyl ethanolamine and ethanolamine. As you can see on plate, we have applied ethanolamine and N-acyl ethanolamine for checking the linearity, and uh, we have generated the chromatograms for the same, and in the chromatograms, as you can see, we got a very good results for the linearity and very good CV value. So by using a CAMAG scanner densitometer, we have scanned the plate under 510 nanometers here. So first of all, after development of the plate, we have captured the plate image. On plate, phospholipids and its impurities are not visible. Then we have derivatized the plate by using dipping technique here. So we have dipped the plate into copper sulfate reagent, and after dipping and heating the plate at 170 degrees Celsius for 10 minutes, the phospholipids bands are appeared onto the plate, and these are phospholipids impurities, ethanolamine and N-acyl ethanolamine. And after development of this plate, we have scanned the plate by using CAMAG densitometer scanner for by using tungsten lamp and 510 nanometer, and after scanning the plate you can see the generated chromatograms of N-acyl ethanolamine. So this is the 3D view of chromatograms generated, and here we have checked the linearity of N-acyl ethanolamine and ethanolamine. And then we did substance assignment, and we got the very good linearity CV value 2.66% and R value more than 0.99 for N-acyl ethanolamine, and here we achieved a very good linearity for phosphatidyl ethanol amine. Generally, as method of HPTLC for phospholipid says CV value should be lesser than 15% in Michaelis Menten 1 regression mode. But here we have used linear 2 regression mode, and in this mode also we have achieved a very good linearity. Here if we apply Michaelis Menten 1 equation, the CV value again going down, and we are getting very good results in uh, any regression mode. And further, software will tell you the detected impurity percentage. But here this analysis we did only for linearity checking. Now I will show you the report generation for this complete analysis. If we are checking any linearity or if we are doing actual batch sample analysis, how report will generate in HPTLC software and how it is compliant with 21 CFR Part 11, I will show you here. So just on a single click here on the report, report includes your sample application sequence, your plate images, chromatograms, automatic calculations, linearity. If you are capture absorption spectra, that spectra will also come in the report. So this is complete analysis report for the linearity checking of phospholipid impurities, phosphatidyl ethanolamine and N-acyl phosphatidyl ethanolamine. The results of linearity with CV value, R value and line equation you can see. You can simply export this report in PDF format and save in your PC. Along with that, you can change the header, you can change the logo, and you can put your industry logo here. You can see the actual plate analysis name along with the creator of the analysis file, its modification done with time tag and date tag, username, everything is reported. And further, if user is having rights of e-sign, we can do e-sign of this analysis by entering our login detail. So this software is fully 21 CFR Part 11 compliant. I'll show you some features in 21 CFR. First of all, we can make different users and we can assign the different rights to different users. So these are nearly 15 rights uh, to give for different users as per hierarchy in your organization, like analyst, reviewer, quality manager, etc. Based on that you can give different rights to users for accessing this software. And here we can set all the password policies, etc. So this software is fully compliant with latest guidelines of 21 CFR Part 11. Here we can assign different e-signature levels and we can see all the logs, whole system log here, and we can trace back any activity. Any activity done in whole system or in a particular analysis file will reflect, and it is though it is compliant, the activity reflect in your audit trail. I'll show you one another example. These are three files I have exported. Here we did actual batch analysis. Total three samples we have applied on a single plate, and we detected only two impurities here. I can put automatic RF values here on the photo documented image of the analysis plate, and I can find out the RF value of separated band easily by using this software, and I can export this image in HD, full HD mode, and save anywhere on our PC. So huge data we are getting from one simple analysis, HPTLC analysis, plate images under different illuminations for R&D purpose or under single illumination as per requirement of QC laboratory, before development and before derivatization images, and after derivatization images, after derivatization with copper sulfate reagent, and then further chromatograms in evaluation tab. We can see the actual chromatograms, area versus RF value response for each phospholipid or its impurity, and further automatic calculations, the linearity, and further quantification. So this is about CAMAG Vision Cat software and its compliance readiness, and how we are doing day-to-day phospholipid analysis in our lab. Now, in this session, I tried to explain you about phospholipids, how different techniques are available, why HPTLC technique is preferable and recommended over other, and how it gives a 5 times faster response with a very less cost per sample analysis and very less time requirement, and how it able to separate polar as well as non-polar lipids, whether it is from natural source or synthetic or semi-synthetic sources. Now the session is open for question and answers. I request you to ask your questions openly. If you want to ask your question, you can raise your hand. Our team will unmute you and you can ask your question live. Along with that, we have our application center. We are routinely dealing with phospholipids analysis. We did several projects for different pharmaceutical industries across India. Top 10 pharmaceutical industries, if I name, those who are making generic drugs and tablet formulations, emulsion formulations, injectables, etc. For them, we are providing complete support of method development and validation. We have installed CAMAG HPTLC systems across India in that premises. Now the session is open for question and answers. Yes.
Hello.
Hello. Yes.
Yes, am I audible?
Yes, sir.
Yes.
Satish Wag, sir. Yes, sir.
Right. Actually, my question is related to the phospholipid fatty acid composition. So, what are the techniques are we going to use to determine the concentration of fatty acid that is in lying in that particular phospholipid? Not I'm specifically, but any of the phospholipid if you take. So, what are the techniques for it? And if you found that composition, then how to analyze it? That's my question.
Okay, that is very good question, sir. Actually, different fatty acid is very big issue. As I shown you in the image, during manufacturing of natural phospholipids, the fatty acids may come along with lecithins. So that fatty acid is a part of oil, and that oil may enters during purification also, and it may generate free fatty acid impurities into the final product of phospholipids. So HPTLC is one of the preferred choice because on the plate, if you apply the main phospholipid substance, then after development of that plate into polar and non-polar mobile phases, which are fixed, as I shown you, we have very good and validated methods available. After separation of that components in your main phospholipid substance, if it is showing only a single band of your product of interest phospholipid substance, then it is okay. But if it is showing any free flowing free fatty acid material, non-polar material on plate in non-polar mobile phase, then we can see that free fatty acid or fatty acid is there in your main substance. So further we can go for scanning densitometry and quantify its percentage. Along with that, any polar impurity is there in your phospholipid, definitely we will apply that phospholipid on plate. We will develop the plate in polar mobile phase, and easily in 1 hour, maximum in 1 hour for 10 samples, we can find out the presence of polar impurities also. So visual detection is the key in HPTLC analysis. Visibly, we can see different impurities, and further we can decide our strategy. by using densitometry, we can do quantification. I hope I tried to give you the
Yes,
complete answer. Yes.
yes, I understand it, but what exactly the limit for that the quantifications?
Okay, so sir, that is a good question because importers are importing phospholipid materials from many vendors. So, as
Mm-hmm.
per vendor specification, as per the purification process, different vendors may can apply different limits for different fatty acids.
Okay,
So we have to check the limit with their standard protocols as per our acceptance criteria, or we can set the limit which will be the gold standard limit as per industry requirement and regulatory compliance, sir. So for different fatty acids or polar impurities, there are different limits, sir.
okay, okay. Thanks
And
for clarity.
yes, yes, sir. And HPTLC can achieve a very lowest limit also. We have detected sometimes we are detecting lesser than 0.1% impurities also.
Okay.
Yeah.
Hello.
Hello, sir. Yes.
I'm not,
Yes, sir. You are audible.
I wanted to ask that why linearity is important to do this analysis. Without performing a linearity, can we not perform the test? Is it mandatory to do so? And is there any statistical method which is saying to perform the linearity before performing the actual test? Is
Okay,
it
okay.
this?
Okay, sir, that is good question. Actually, there are two types of analysis, qualitative and quantitative. In qualitative analysis, we can screen the large pool of samples in rapid way by using HPTLC. If you want to do further quantification, in-depth quantification, we can use multi-level calibration curve method. And for using multi-level calibration curve method, we can plot the linearity for accurate and sensitive quantification. In CAMAG HPTLC software and in HPTLC system, we are generally recommending to perform a multi-level calibration curve quantification from samples. So initially, for by using
Means it
qualitative
is
method,
not from mandatory from the
no, no, sir.
hello.
For
Means sir,
method
it is not mandatory from
mandatory. Yes, it is not mandatory, sir. If for large sample screening for qualitative analysis, linearity checking is not mandatory, sir.
we are performing to just verify how the instrument is performing before the test, isn't it?
Sir, if we are doing any quantification, so for quantification studies, we are performing linearity and the quantification methods as per ICH Q2 R2 guidelines, we have to prove its linearity. For method validation, definitely linearity is required, sir.
Is there any calculation with the linearity?
Sir, software will do automatic calculations, so no need to worry. Software will automatically calculate, and it is ISO certified software. So the CV value generated, coefficient of variance, and correlation coefficient R value are very much reliable, and you can cross check with your manual calculation methods. But software will take care of all the calculations. You can directly report that generated data and values.
Understand that software will calculate all the things, but I'm saying that is there mandatory to calculate linearity or the RSD like it's because sometime in HPLC we have to calculate your RSD before performing the actual essay. Then, like I'm asking
Okay,
that
okay, sir.
is it mandatory
Correct.
to calculate that things? My point is
Yes, sir.
that.
It is required, sir.
Okay, so there are multiple you have shown that multiple impurities are present there. So linearity we are performing for the all of the impurities
Uh, no, sir. Only
or
the
only one
impurities.
we are
No, no,
performing.
sir. The impurities detected in your phospholipid. Only for that we are performing linearity. Okay, sir. So there will be many
So
impurities in your
phospholipid
sample.
concentration we are taking. I'm
Hello.
asking, sir, that we are taking the phospholipids at multiple dilutions, and then we are detecting its linearity, and against that we are just checking, isn't it?
No, sir. Initially, we are doing qualitative analysis. We are applying multiple samples on a single plate, and after knowing the composition of that phospholipid, if it is impurity free, there is no impurity detected in initial screening, then no need to go for its quantification. But if any impurity is detected in your sample, then you can apply multi-level calibration curve and do the quantification. It is that simple. First of all, screen with qualitative method, same method, and if any impurity detected, only for that particular impurity you have to apply linearity.
Okay, so means a single unity, all the RF and other whatever will the come at the later stage, that impurity will be calculated, isn't it?
Yes, sir. Any detected impurity in your sample, we can further quantify by using scanning densitometers.
Hello, good afternoon.
Okay, thank you.
Okay, thank you, sir.
Hello.
Hello, good
Hello.
afternoon.
Yeah, good
Hello.
afternoon, sir.
Sir, I have one doubt regarding you say now this copper sulfate solution may be used for the derivatization. So,
Yes, sir.
which solvent we can useful, either organic or water or no aqueous, which solvent?
Sir, we have used water, aqueous copper sulfate.
Copper sulfate, okay.
Yeah, but by using CAMAG immersion device and or CAMAG derivatizer, we are not facing many problems, because if we do manual derivatization,
Yeah, yeah,
it is problematic.
yes, sir.
Water is highly polar and silica is highly polar.
Yes.
So we need to be careful during the analysis, but we are using instrumentation, then it is very easy and
Very
fast.
easy. That's why what are the other phytomarkers we are doing? Dipping is in manually method. There is no problem. But in his water is containing that time we have that the drag type of peaks are coming sometimes.
Okay,
Those
okay.
who are like interacting with water. So in this manual method, any other techniques, any tricks will be available. Can you suggest it for that?
Sir, instead of dipping, there is one other method of spraying the deratizing reagent on plate. For that, we have CAMAC derivatizer for uniform and user-independent derivatization. So that method is available with us for analysis of phospholipid.
Okay.
We will share with you, sir,
Okay. Sir,
on
one
your
more
email
query
ID.
regarding in that. Sometimes we are doing this phospholipids. Either we need to do like what can call recovery studies or intraday, interday. Those are ICH Q2 guidelines, and it will be not required.
Okay, sir. If you are developing some new methods, then validation is required. If you are using already validated method from any pharmacopoeia or any compendium, then it is not required to validate again and again. You can
Okay,
utilize it as it is in your lab.
okay. Sir, second one thing, last question. My like I prepared one sample concentration like 100 mg per ml, but my linearity curve is going for 100 to 1000 nanogram. But my
Okay.
sample is not detecting on that linearity. But linearity CV and the percentage is very good. Shall I accept those method for further process? Either what are the sample I selected that should be and comes under the linearity curve.
Sir, it is depend on aim of your analysis. If you want to exactly quantify the amount of that particular impurity,
Yes.
then you have to come with the conclusion in quantification. You have to dilute the sample to so that the impurity in your sample will come in the middle of linearity. But if it is passing the limit, it is lesser than your given limit, then no need to, for limit test, no need to quantify.
Okay. Okay. The quantification should be not under the linearity of the particular concentration. So,
Yes.
thanks. Thank you.
I think in question and answer box there are uh, some questions. Please disclose the type of TLC plates you are using for polar and non-polar phospholipids. So generally we are using same TLC silica gel plates coated on aluminum backing material or glass backing material. Any more question live? Anyone wanted to ask question? Please unmute yourself and ask.
Hello, good
Shiv-
afternoon.
Yeah, good afternoon, sir.
Yes, sir, can we send our sample to your lab for analysis purpose so we can confirm our results?
Open tab Q&A.
Hello.
Yes, sir. Hello,
So
sir.
can we send our samples to your lab for analysis
Uh,
so we can confirm our
Yes, sir. Yes, sir. Our lab is open for sample analysis related to phospholipids. I request you to send the email with your aim of the analysis and institutional details on lab@enchrome.in. lab@enchrome.in.
Okay. And currently we are using the reference standard of Merck, in which we are getting, it is a mixture of phospholipids and we are getting the 5 peaks.
Okay.
And we are quantifying with respect to the area, not with the linearity. So it is okay.
Yes, sir. Yes, sir. Linearity we are generating with respect to area only, sir. And single point calibration also we can perform, sir. As per your method stated, we can do the analysis.
Okay. And regarding the copper sulfate, can you tell us how to prepare the solution?
Okay, sir, copper sulfate along with, first of all, we have to dissolve the copper sulfate in phosphoric acid, and then we have to add a remaining portion of the water into that. So the preparation recipe is given in CAMAG application note also. We will share that complete phospholipid analysis method with derivatizing reagent preparation with you, sir.
Okay, thank you. Thanks a lot.
Hello, yes, ma'am, you are audible.
Sir, actually, I am Dr. Shilpa.
Okay, yes, ma'am.
I would like to know why.
Ma'am, your voice is very much means there is interference. So I request you to join only from one device or hello. Your voice is echoing.
Hello.
Ma'am, you can type your question in Q&A box so that he'll answer.
Okay, fine.
I think whether we can analyze the concentration of radio labeled drug. Ma'am, actually, for that, we have many different pharmaceutical applications in HPTLC. Radio labeled drug is entirely a different kind of topic. So, we will arrange a separate webinar or we will discuss separate in later. You are interested in that drugs analysis, we will share the available methods, if any, or we will share the possible aspects of HPTLC with you, sir, with you, ma'am, on this email.
Can I ask my question now?
Yes, ma'am.
Okay, my question was that what I understand is you have been focusing on QC of phospholipids, which are used as excipients. Can we utilize a similar method for quantification of these fats which are coming from natural origin, seeds or plant material? So, can we extrapolate this method for the same?
Yes, ma'am. I have shown that method also, quantification method. I have shown complete quantification of different fatty acids like tripalmitin or stearic acid, etc. Non-polar phospholipidic impurities I have shown. We can do further quantification anytime, ma'am.
Okay, but then if the fatty acids are of almost similar molecular mass, then it would be a challenge for first separating them and then doing quantification with respect to a marker. So, have you done any?
Molecular mass will be same, but their polarities and chemical properties may be different. And HPTLC is very selective with respect to polarities. So we can separate because I have seen the separation of different fatty acids from fixed oils. Okay, ma'am. So we can, if method available, we will search for that. So this current method also we can utilize. But if any new fatty acid come in picture, we can try to develop method on HPTLC.
Yeah, okay, thank you.
Because this HPTLC technique is matrix robust any sample matrix containing any kind of interferences, etc., we can first try HPTLC method development.
Yeah, right. Yeah, thank
Yeah,
you.
thank you, ma'am. Pallavi, ma'am, ask one question. Sir, how we validate linearity specificity of the sample during HPT? It would help us a lot. Ma'am, we have delivered one webinar in the month of December 2025, I think. We have posted that HPTLC method validation, a complete webinar on our YouTube channel. I request you to please subscribe our YouTube channel, visit our YouTube channel, and search for method validation in HPTLC field. Please provide information about sample preparation of phospholipids. Okay, so sample preparation in phospholipid analysis is very easy. If we uh, are doing analysis of non-polar phospholipids, we have to dissolve the uh, phospholipid sample in non-polar solvent that are given in our method, CAMAG method, as well as Anchrome in-house developed method. Or if we are dealing with polar phospholipids, we have to dissolve in polar solvents. So sample preparation is very easy. Just dissolve the phospholipids, sonicate for 5 to 10 minutes. And if it is appearing a clear solution, then go for directly application on plate. Sir, while quantifying a herbal extract, we observe variation in the results. Ma'am, actually for herbal extract analysis, we will provide you a separate application support. I think Shubhangi Mehra ma'am asked this question. So we will provide you separate support on call. Thank you, ma'am.
Good afternoon, sir.
Yes, sir.
Sir, what is the minimum concentration which is quantified for these impurities, phospholipid impurities, if you said? Because these will be available in traces in the formulation if you go for.
Yes, sir.
So,
Sensitivity of
for that,
CAMAC. Yes, yes, sir. Sensitivity of CAMAC scanning densitometer is up to nanograms for UV and visible components. So phospholipids are visible under white light, so we can go and achieve up to nanogram level detection.
okay. One more thing, sir, I wanted to ask because whenever we go for derivatization with any derivatizing agent, okay, so that repetition, like repetitive thing, precision we are not getting. So, as you suggested that derivatizer you are supposed to
Okay.
use, but if
So
we don't
with
have that
respect to
for derivatizer,
precision, generally if we use applicators, CAMAG applicators like ATS4 or Linomat 5, completely automatic and
that type.
semi-automatic, definitely it will apply exact volume and you can achieve uh, uh, band-to-band area reproducibility and repeatability. Definitely, sir. And if you
And
follow yeah,
the last thing which I want to understand is the dipping time you said in the USO4 policy, right?
yes.
So, how much time we are supposed to hold the plate or dip the plate into the solution?
As per our practical experience, sir, only for 1 to 2 seconds we have to hold the plate into copper sulfate reagent, and then we have to take out the plate immediately. And after taking out the plate, wipe out the back surface of the plate and just wait for 5 more minutes, and then keep the plate on CAMAC plate heater to heat the plate at 170 degrees Celsius.
170 degrees Celsius. Can we keep it in oven as well?
But oven will not provide you the homogeneous heating of each bed on the plate. Uh, uh, CAMAC plate heater is very well designed for heating the plate TLC or HPTLC plate, either
But
aluminum backing or a glass backing material.
what if we don't have that?
You can
Because we
utilize
have
oven,
lot
but the
of
temperature should be well calibrated.
okay. Air drying will not do.
No, dryer is not for heating purpose. Dryer is for drying purpose. We have to use a different heater like oven or CAMAC plate heater for
No,
heating the plate after
170 is the
derivitization.
optimized temperature you said.
Hello.
170 is the optimized temperature for the same.
No, 170 is for phospholipids analysis. And for different, if you are dealing with herbal or other phytochemicals, there will be different.
I'm asking for phospholipids only.
Phospholipids 170 degree for 10 minutes, sir.
Okay, fine. Thank you, sir.
Okay, okay, sir.
Yeah.
Okay, there is one question from Kamal Joshi. Can we quantify the single analyte like LPC or PC by this method? Yes, lysophosphatidylcholine and phosphatidylcholine we can analyze and quantify. The please share full method documents. Yes, we will share with you, sir. Phospholipids can be visualized. Yes, phospholipids visualize as I shown you in methods. Then sometimes the plate become black after derivatization. So due to manual derivatization technique, it may happen. But if we prefer CAMAG immersion device or derivatizer, this problem may not arise. How to decide composition of mobile phase? Yes, definitely. Depend upon polarity. Polarity plays vital role. Can we send sample to your lab for analysis purpose? Yes, you can send your samples for analysis purpose. But before that, I request you to connect with us on lab@enchrome.in. That is our uh, email ID on which you have to send the aim of the analysis, then your contact details, industry details. Then depend on feasibility of the project, we will do the analysis. I think I tried to give answers of many questions in Q&A box. Those who are related to the our topic of interest today, phospholipids analysis by HPTLC. So I request you all to join our upcoming webinar also. Thank you so much for joining this session with the Enchrome team and knowing more about phospholipids. Uh, Thank you so much for joining. In next upcoming webinar, yes, in next upcoming webinar, we will discuss about EG-DEG analysis, ethylene glycol, diethylene glycol analysis from syrup samples and how HPTLC gives you the solution to detect this unwanted contamination and very hazardous contamination in syrup samples. This is also useful for pharmaceutical as well as syrup manufacturers in herbal industry also. Thank you from Enchrome. These are our contact details. And at the end of this session, we will again provide you one poll question that will be a feedback kind of question. I request you to answer that question also. The remaining questions in Q&A box, we will try to answer all the questions and provide the corresponding documents to that concerned person on their email IDs. Thank you once again for your valuable time and attending this session.